So that's why we have the 23 pairs. So we have the 46. In meiosis 1, then, we're going to split that in half. Okay, so we've doubled it. So do you remember when we did 2A, there was the cell cycle that we looked at and we divided it into G1, X, and G2, okay? So during G1, it simply grows and it's getting prepared. S is the synthesis phase, where it's going to duplicate the DNA. So when this started off, it really had 92, 92, I did the math right? So it's going to split in half and split in half again. So it's one duplication event, just one S phase, but two consecutive divisions.
Do we need to know what happens in S phase?
Yes, we have to know. Bottom line is this: you start off in meiosis. You start off with the diploid, right? And you're going to end with 4 haploids.
That's not just first.
No, that's also for the egg as well. The egg just doesn't finish. Now the egg, if you're talking about how many you end up with, you're right. With the egg, you'll start with 1 and you'll only end with 1. And that has to do with the fact that the egg, if you looked at it under the microscope, all the students want to say that the nucleus is the cell. It is huge. It is the largest cell that we have in the body. I mean, it is so long and big that what has happened is that all 4 of them contribute to just one cytoplasm, so that it can stay that big. So the other 3 have to die off, and it's actually going to die off at this stage in the female. Okay, so and then the other one's going to stay. So we get these what are called polar bodies. So you don't get it. But yes, the idea of general meiosis is you start off with 1 and you end up with 4. And the beauty of that meiosis is that obviously it's different what you began with, what you end with, than what you began with, right? Because you've got half of what you started off with. But also, between all 4 of them, there's been genetic shuffling. So we've had this crossing over. So before this even occurs, at this stage, chromosome 1 from my mom and chromosome from my dad are going to cross over, and they're going to share genetic information. So that the 23 that we have over there are different between those 4 and completely a different shuffled up deck of what we had from this one that we originated. Because if you remember mitosis, we started off with 1 cell and end up with 2 cells that were identical between them and identical to the parental. So here we've got this variety created, where we've got this idea of we now have 4, and there are 4 different cells, and they only have half. Why do we need half? Well, because we're going to have sexual reproduction, okay? We're going to have these chromosomes coming from the male and these chromosomes coming from the female. If we bring every time 2N together, then I go to 4N. And then if I bring 4N and 4N, I'm going to have 8N. I'm going to have this huge amount of genetic information. Okay, that you can see that even if you have, 1 extra pair, it's not going to live. So the reason we do split it in half is so that we can bring it together and get all this variety, which is exciting.
Can you go over the part, which happens at which phase?
Oh, okay.
So you said we didn't need to know phases.
No, you don't need to know the phases. I won't test you on that. I just want you to have this idea that when we talk about meiosis, we have 1 duplication, okay, but 2 divisions. The 2 divisions are called meiosis 1 and meiosis 2. And each one has the same exact steps as mitosis: prophase, metaphase, anaphase, telophase. So from here, we're going to have our first division, okay? The first division is going to go through a prophase, metaphase, anaphase, telophase, and now I get the haploid. So I've reduced it in half. Now I'm going to get, from these 2, yet another division. That's a duplication event, okay? So I have it duplicated, and now I'm going to end up now with half of what we had when we began, okay? That's it. That's what I want to know.
Does that relate to that?
And now this is related to that, yes. So in the male, what's going to happen? The male is going to do this exactly the way it says. And it's going to do it in trillions, okay? Basically from sexual maturity on, millions and millions of these are produced a day, okay? Every ejaculate has what, 10 million sperm in it? I mean, it's just this constant production. So it goes from 1 to 4, and how does it do that? It does it in the testes and the seminiferous tubules. So when you look at it histologically in the tubule, okay, so if you have the tubule that's going like this, you're going to have all these cells, okay? And what they're going to do is they're going to be undergoing meiosis from the edge and work their way inward, okay? So that by the end, you've got the little sperm that are in the lumen of that seminiferous tubule. So you start here with the spermatogonia. So that's this. We call this cell the spermatogonia in the male. We call these the primary spermatocytes. We call these the secondary, and now there's maturation events. it has to grow a tail, it has to be prepared. So what's going to happen really with the male sperm is the female is going to have this huge egg that has lots and lots of cytoplasm. The male is only going to provide its nucleus, its DNA. So its job basically is to get to the egg. So it's going to have a head, and you're going to have all this, you're going to look at that for the lecture, and it's going to have, right here at the base of the tail, it's going to have lots and lots of mitochondria so that it can move. And while it's in the male reproductive tract, that tail is not working yet. You don't want it to waste its energy here. It's got to waste the ATP to move in the female reproductive tract. And basically it has to go from the vagina where it's deposited all the way up, and a proper fertilization should occur in the ovarian tubes, in the fallopian tubes, okay? So it's got to go all the way up, okay, and make its way in there and find it. So that energy is going to be stored. So the spermatogonia is going to start on the edge, and here we also have the sustentacular cells, okay, on the edge, and those I do expect you to identify the difference. So you're going to have these cells. The sustentacular cells stay on the edge, so they don't move. They don't divide and migrate inward. They're always on the edge. They've got a nice normal nucleus, whereas, so that one would be sustentacular. Their job, basically, is to produce testosterone to stimulate all of this process, but to produce what's called a blood-testis barrier. They are not going to allow for the things, the toxics that are in the chemicals in the blood. You want to make sure they don't make their way into the germ cells, into these cells that are ultimately going to create the new offspring. So they're pretty protected, just like the brain. So we're going to have that type of a barrier that we can see. When we look at it, the spermatogonia are going to have very dark nuclei, because if you remember in mitosis, before they undergo mitosis, they actually have to condense the DNA, and you can see that histologically when you're looking at it, okay? So in the male, it's going to go just like this, okay? And you're going to look at the steps, and each step is going to have a name. So for example, at this point, this would be sperm. Once it's mature with the little tails, that's going to be sperm. But there is a step before it, which is the spermatid, and that's simply just preparing itself. It's the tail, it's acquiring, making ATP to store, and actually making the mitochondria to be able to do that. So you see that there are steps in between. But the idea then is that the female is going to start off with the oogonia, okay? And she is going to actually be born with the amount of eggs that she will have in her life. So the male is producing these, trillions. And this is why contraceptives target the female. The female is only going to release, theoretically, 1 egg per month. So obviously it's easier to target and prevent one from the millions, okay? And I try to justify that in my mind with my husband, but it makes sense. I mean, if you're gonna stop somebody, it's easier to stop one than millions that are coming out. Yes.
Okay, if the 46 is the spermatogonia,
yes.
Then which one's spermatocyte?
The primary spermatocyte, I believe that would be this one, because there's actually 2 stages.
And where's the secondary one?
Where's your textbook? Let me make sure I'm not talking silliness, okay.
No, I missed it.
Because there actually is 2 stages